One advantage of this protocol is that the worm sheet is easy to handle. The sheet has features that prevent drying, enabling us to conduct microbeam irradiation to living animals, perform several behavioral assays, and long-term observation. The microfluidic channels in this worm sheet are open, making it easier to collect animals. These worm sheets can also be used repeatedly, making them more economical.
When we were developing this technique, we wanted to irradiate only a part of the animals with a microbeam under a microscope. So we needed clear, very thin chips capable of holding the animals.
To begin, place a thin, transparent sheet onto the stage for use as a bottom cover film. Gently position a worm sheet on the bottom cover film with flat tweezers. Place at least 3 droplets of buffer solution on the surface of a 6 cm non-treated Petri dish. Then pick several adult C.Elegans with a platina picker from a culture plate. Transfer the animals to a droplet with the platina picker and allow the animals to swim in buffer to remove any bacteria. Wash the animals in 2 separate droplets and rinse the animals in another droplet. Next, place a droplet of buffer solution onto the surface of the worm sheet. Remove the washed animals from the buffer droplet and transfer them to a droplet on the worm sheet. Use flat tweezers to place a PS cover film over the worm sheet and press gently over the channels from one end of the sheet to the other.
I'll simply drop the animals into a droplet and gently seal with a cover film.